I'm Bruce Gellin, the president of Global Immunization at the Sabin Vaccine Institute. And on behalf of the Sabin Vaccine Institute, I'd like to welcome everyone to this 10th International Conference on Typhoid and Other Invasive Salmonelloses. That over 300 people have committed to coming here to work together on this problem is a testament to the importance that each of you see in moving from evidence to action, the theme of our conference. Special thanks to the Bill and Melinda Gates Foundation for their generous support to make this happen, and thanks to the Uganda's Ministry of Health for their support in welcoming us to their beautiful country. As you all know, this conference is being held in an important and momentous time for the typhoid community. The theme of this conference, as I mentioned, is from evidence to action. So let's talk about both, now and throughout the conference. The evidence. A perfect storm is brewing, and there's an urgency for prevention and control. Rapid urbanization is leading to large numbers of people living in close proximity with poor water and sanitation infrastructure. Climate change is making already vulnerable communities even more vulnerable to disease due to dangerous weather patterns that damage fragile infrastructure and threaten food and water sources. Globalization is connecting populations across the world, giving pathogens opportunities to easily travel, and migrations of people add a new dimension to the mixing of people and pathogens. And antimicrobial resistance, a broad global threat, is especially worrisome in our field. In February, the World Health Organization includes Salmonella on its list of 12 antibiotic-resistant priority pathogens that pose the greatest threats to human health. Multi-drug resistant strains of Salmonella typhi, which first appeared in the 70s in Asia, have now spread to Africa. And resistant strains like Salmonella typhi are leading to more intensive treatment and an increase in the economic burden on both health systems and families. But this has now become an urgent issue. We're facing a post-antibiotic future when typhoid infections won't be difficult to treat, they will be untreatable, as untreatable as they were in the pre-antibiotic era. When we look back to these swirling clouds of this coming perfect storm, we'll look back to this meeting and the actions that it inspires. Over the next 3 days, experts from around the globe and from a wide range of disciplines will be sharing some of the latest data and leading discussions. So please keep the theme, evidence to action, in mind at all times while you're here, today and throughout the conference. On our final session on Thursday, there'll be a time to reflect on all that we've learned to help shape the path forward. And to be sure that we get all of your best ideas, also keep an eye out for 3 surveys that will be distributed throughout the conference. And as you all know, incentives inspire action. So there's an incentive for participating in the surveys, and there's a small cash prize for your participation. We'll announce the winners at the end of each day, and like all such contests, you must be present to win. So thank you for coming, and I'm honored to introduce my colleague Denise Garrett, Vice President of Typhoid Programs at the Sabin Vaccine Institute, to tell you how the rest of the 3 days will unfold. Denise, thank you.
Thank you, Bruce, for the introduction, and welcome out to the conference. I have communicated through email with most of you, and I'm excited to have you here for the largest conference on typhoid ever hosted, with more than 300 attendees representing 45 countries. So we have 45 countries represented in this room. We started working on this conference almost 2 years ago, and we couldn't have done it without a great scientific committee and abstract review panel that helped us curate this comprehensive and inspiring agenda. So can the members of the steering committee and abstract review panel, there are here, please stand. Yes, there are more. All right. Yes, we would like to thank you very much for your contribution to make this a success. Thank you. We are truly embracing the conference theme, evidence to action, from evidence to action. So the agenda is complete with the newest data, along with strategies and policies to turn that knowledge into results. Over the next 3 days, we will be covering the latest burden on typhoid, paratyphoid, and invasive non-typhoid Salmonella, the newest innovations in diagnostics and treatment, the most effective strategies to manage outbreaks, the impact of growing antimicrobial resistance, the new vaccines in the pipeline, including a whole session dedicated to the typhoid conjugated vaccine. And we will wrap up the conference with Mission Possible, From Evidence to Action, a session highlighting upcoming policy decisions that will mobilize our research and translate it in prevention and control measures. Before we get into the first session, I have a few housekeeping points for the conference. Posters will be displayed upstairs throughout the whole conference. The presenters will be standing by their posters to discuss their work with you during the afternoon coffee breaks today and tomorrow, and then on Thursday during the morning coffee break. Coffee breaks and lunch will be served upstairs. And if you have any questions, please find one of the event staff. There is also a conference office that's located next to the plenary hall, and you can find someone there during the conference hours. Finally, after our program today, we have a welcome reception featuring remarks from the Honorable Dr. Jane Esang, the Uganda's Minister of Health. Uganda has been such a wonderful host country for this conference, and I'm thrilled that the minister will be joining us tonight. I hope that you will find the 3 days of the programming that we have prepared for you, as well as networking with your peers, a valuable experience. So now, for our first stimulating session, I will turn it over to our session moderators, Thomas Cherian and Melita Gordon. So please sit back, relax, and enjoy the conference.
Thank you very much. Welcome everyone to our session on epidemiology and global burden. I'd like to straight away invite Anita Zaidi to come up and start. Anita, many of you will know, is director of the Gates Foundation Enteric and Diarrheal Diseases Program. Welcome, Anita. Thank you.
Thank you, Melita. It's a real, I'm really delighted to be here. It's been a journey that I wanted to share with you today at the foundation on typhoid. And let's see, I have that. And the theme is evidence to action, which is how we've been thinking about typhoid at the foundation. And so I look forward to sharing that with you. Let's see if I can get this to work. And so maybe it is, yeah, so you're correct. It's a little counterintuitive because it's down is up. So in November 2015, we had the opportunity to present to our co-chairs our new thinking about typhoid. And the argument that we made was that this is quintessentially a disease of the poor, that this was a neglected tropical public health problem, and that there was a possibility to do much more than we have been doing to accelerate the field and accelerate impact. And the main arguments that we made were around AMR. So typhoid, I think is probably the most dangerous community pathogen from the point of view of antimicrobial resistance and running out of treatment options. We also made the point that water and sanitation, although historically have been a really fantastic way of controlling typhoid, may not in the short to medium term yield the benefits that we want to see for typhoid. And now one of the things that the foundation likes to do is find areas where you can be catalytic, where you can, some amount of funding can really make a huge difference to impact. And so we thought that typhoid really fit that sweet spot to be able to do a lot. And so this strategy was approved, and we've been rolling it out slowly. But this is my first opportunity to actually share it with the typhoid community. I have presented this before at the Drop Med, but not all of you were there then. Let's see. There we go. So our strategy is oriented around impact, and we chose in 2015 a 2035, 20-year timeline to get to aggressive typhoid control. We did not, and many of you in the room will remember, we went through a series of consultations in 2015 to see where we should be going. Should we think about eradication for typhoid? Is that too bold? And think about elimination, etc. And all of that thinking was very useful in formulating our work. All right, so here's what we are doing. We have 6 areas of work which we formulated for 2020 goals, but these get us to pathway to impact by 2035. And the areas are having a really good vaccine. So we are committed to having 2 pre-qualified vaccines by 2020. We wanted to do all of the work that would be necessary to get the appropriate recommendations from SAGE and from Gavi for financing. We believe that we will need next generation Salmonella vaccines, so we need to start doing the work for those right now. We also think that we need to see where the Foundation's WASH strategy could be useful for typhoid control, and to demonstrate by developing a toolkit how one could have maximum impact. And this refers to there are geographies where the Foundation works at the municipal level or the medium intermediate level for waste disposal, for waste treatment plants, etc. And then to also look at what do we need to do to understand transmission modeling, and could we look at potentially elimination as a possibility. And as you can see through the boxes on the right, there is an aggressive timeline to get the 2020 work done, but there is thinking about 2035 and how do we then get into beyond the 2020 period, maximizing coverage of interventions and getting towards sustained reduction in goals. So our body of work relates, went the wrong way. These goals relate to the work that we've been funding. And of these, there's actually, since 2015, there's been substantial progress, and many of you are grantees, and this is an opportunity to kind of see how you fit into the big scheme of things. So, continued TCV development efforts, these are mainly with IVI. Early development of new generation broadly cross-protective vaccines, many of you are working on this. Strategic lead partner to accelerate vaccine introduction, and that's the really amazing thing that we've been able to accomplish together with you, and I'll talk about this a little bit more. Then effectiveness of a toolkit, and this incorporates also WASH, and then there are some underlying studies that we are trying to understand right now related to transmission. And then the last but not least is sort of the fundamental biology of typhoid, understanding carriage, if we were to have an elimination goal. So I put this because of course we can have great ideas, but nothing will happen without all of you. And this is, for many of you who have visited the foundation, this is a proverb that's right up there as soon as you enter the conference building. It's if you want to go fast, go alone, but if you want to go far, go together. So we can have fantastic goals, but without you, nothing is possible. And especially the early part, up to the 2020, but then as it goes forward, all of you, each one of you will need to be a champion to get the work done. And a beginning has been made with this TIVAC, Typhoid Vaccine Acceleration Consortium, which brings together University of Maryland, Oxford, and PATH to reach out to many of you to generate the evidence that we will need to have for country adoption, for all the right recommendations and for country adoption. And, yeah, I have the animation, which is not working. Okay, there we go. So what I really liked about the TIVAC is it has the word together in it. So this is the hashtag, yeah, there we go. This is the hashtag, and it's, together we can take on typhoid. And there is a reception on the 5th, which I think Kathy has sent out an invitation to everybody. Great. Okay, so switching gears, there is another pretty interesting development on the horizon. And this is taken from The Economist of the 23rd of March, and it's talking about what's coming. And what's coming is hopefully a data revolution in scientific publications for global health. And what this is a portal that's being established along the lines that Wellcome has already done, called Gates Open Research. And this will allow you to publish your studies that are funded by Gates, and it will appear as a publication within 7 days of publication, and then there will be an open review process, which you can, as long as your reviewers, suggested reviewers meet criteria, you'll be able to see what they say about your thing, and it's up to you then whether you revise it or respond to the reviewers' comments, etc. So the hope is that this will really accelerate the timelines for publication of global health. And for a community such as typhoid, which is small but growing, it could lead to impact much quicker than in the old way of doing things, where you don't see stuff 2 to 3 years after it was finished. So I'm very excited about this. I think it's also an opportunity for developing country scientists to get their work into a place where it will be read widely. And we all struggle through publishing from developing countries because we can't get the journals to take developing country scientists seriously. And so I think there'll be time for questions, so I can help answer those. And there is a URL that you can go and start looking at this. When will this become live? It's actually happening soon. We expect it to be active by late summer. So if you have stuff that you already want to submit, that'd be great. Thank you.
Thank you very much, Anita. And just as Anita says, if you could keep your questions till the end of the session, we'll have an open session of questions with the panelists. Thanks, Anita.
Thank you very much, Anita, and I have the privilege of introducing the next speaker who will make a presentation on the evolution of the epidemiology of typhoid fever, John Crump from the University of Otago, also known in the typhoid circles as the John Crump.
Great. Thanks for the introduction, and thanks for the opportunity to speak. And I'd have to say nothing quickens the mind more than having to speak to a group of respected colleagues who know typhoid in and out. So I'll spend the next 15 minutes touching on some progress and also some challenges in typhoid epidemiology. Of the many areas of interest in typhoid epidemiology, I've picked some aspects that I think are quite topical, although perhaps not the most obvious. And I sort of cringe a little bit to do this, but although seemingly fundamental, I'll discuss mode of transmission and sources of Salmonella typhi, reviewing current evidence and the central importance of work in this area for designing non-vaccine prevention interventions. And then I'll move to progress with burden of disease efforts, looking at the current estimates and also what we've seen in trends over recent years, including a discussion on extrapolation, uncertainty, data gaps, especially the challenge of understanding severe disease and deaths, and particularly the issue of typhoid occurrence by age, which is particularly relevant to conjugate vaccine policy decisions. So this slide gives an epidemiologist's perspective of the major classes of modes of transmission of an infection with humans as the main reservoir. We think of direct transmission as the collective noun for transmission by direct contact or droplet spread. And we tend to equate direct transmission with the term person-to-person transmission, which in its strictest sense is transmission by direct contact between persons without intermediaries, as is, say, the case for Ebola virus disease. And I bring this up because this is a term that's understood differently by people in this audience, and so I thought it was important to highlight this. The other major class is indirect transmission and includes airborne, vector-borne, and vehicle-borne modes of transmission. And for the most part, we think of typhoid fever as predominantly indirectly transmitted, with the vehicles being food and water often. The reason for belaboring these points is that mode of transmission is of great importance to the design of non-vaccine prevention interventions. And without a robust understanding of mode of transmission, the prevention intervention menu is extremely diverse, running the gamut from full body personal protective equipment to interrupt directly transmitted pathogens, to improving the microbiologic quality of drinking water for pathogens with a water vehicle. I'd point out that transmission-based hospital infection control guidelines reflect the perspective illustrated here, recommending standard precautions for patients with typhoid and only invoking contact precautions for diapered or incontinent persons for the duration of illness or to control institutional outbreaks, and not invoking droplet precautions. So putting the chain of infection together for Salmonella typhi, the epidemiologic definition of a reservoir is the habitat in which the agent normally lives, grows, and multiplies. And of course, our current understanding is that humans are the reservoir of Salmonella typhi. Of course, human reservoirs may be in different infection states, ranging from acutely infected to chronic carriers. And at present, the portal of exit for the pathogen is fairly uncontroversial. The source is defined as the point at which the agent is transferred from the host. And for typhoid, we think about water and food as important sources, but of course the reservoir may be a source, as has been demonstrated in incidences of oral anal sexual transmission of Salmonella typhi. We think of the mode of transmission as involving water and food vehicles, but reserve an other category, which I'll come back to. The portal of entry remains fairly uncontroversial through the mouth, and the susceptible host is another human. And I'd like to comment briefly on a couple of questions asked by epidemiologists in both epidemic and endemic typhoid situations. The first is, what is the source and predominant mode of transmission of Salmonella typhi, both in general at the global level, but also in this particular epidemiologic context, because it can vary widely. Understanding this is vital for targeting non-vaccine prevention interventions and varies considerably from place to place. And I think a second area of great interest to us as a community is what's the relative contribution of the infection classes contributing to shedding during acute infection versus chronic carriage to onward transmission. So what have we learned about mode of transmission in aggregate since the last meeting? Well, I'll highlight a couple of contributions. Mogasali and colleagues published their systematic review and meta-analysis of the association between unimproved water and typhoid fever risk, showing that across eligible studies and controlling for other factors, persons with unimproved water had 2.4 times the odds of typhoid fever compared with those with improved water. And I do think aggregating studies in this way is quite helpful, and in this case quite reassuring. And then I wanted to also highlight a paper that we might have missed, which is the World Health Organization Foodborne Disease Epidemiology Reference Group Source Attribution Task Force paper that's recently been published following an expert elicitation from 72 enteric disease experts from all WHO regions using Cook's classical method. And across regions, the solicitation identified the contribution of waterborne transmission as accounting from 0.33 to 0.57 of disease, foodborne transmission 0.08 to 0.49, and other transmission 0.11 to 0.47. And this paper is quite rich and well worth reading to appreciate regional differences in perceived predominant modes of transmission of typhoid. As always, there's been a lot of work on studies examining sources and modes of transmission of endemic and epidemic typhoid in specific localities. And to illustrate the diversity of what goes on in typhoid epidemiology, we continue to see documentation of foodborne transmission in the form of restaurant-associated outbreaks linked to chronic carriage and food handlers in countries where typhoid's uncommon, like the outbreak recently reported from Colorado in the United States. We've seen examples of wide-scale waterborne transmission, such as the detailed study of Kariuki and colleagues implicating municipal water supplies in Kathmandu, Nepal, published last year. And also situations where both water and food vehicles are important, such as the work in Central Division, Fiji, by Prasad and Jenkins, presented in final form at this meeting, associating disease with both surface water consumption and also the use of unwashed produce, often grown in pit latrine seepage. So I just wanted to point out that this kind of bread and butter epidemiology remains invaluable to inform priority measures for disease control in specific locations. So let me just turn briefly to now infection class and transmission. As a community, I think we classically think of, and probably simplistically think of, typhoid fecal shedding in the classes of acute disease, with shedding less than 3 months after onset, convalescent carriage 3 to 12 months after onset, and chronic carriage 12 months or longer after onset. And of course, we'd like to know much more about how each class contributes to ongoing transmission in different settings. On this topic, some things that we think we know include that at lower force of infection, a larger proportion of acute typhoid cases occur in age groups that are more susceptible to becoming chronic carriers. And as illustrated in the Colorado restaurant outbreak, acute typhoid infections contribution to onward transmission approaches zero as acute disease approaches zero. And so in low incidence settings, chronic carriers probably contribute close to 100% of ongoing transmission. Without going into detail of specific questions, we've got much to learn about the contribution of infection class to onward transmission in different settings. Now let me move on to burden. Need to go back a slide. Right. In this slide, I've tried to summarize the history of published estimates of typhoid burden. This slide shows the year of the estimate as columns from oldest to most recent. The origin, coverage, and burden metrics are shown in rows. And I'll show you these data visually soon, but I wanted to point out that the number and frequency of published estimates has been increasing pleasingly. And without going into details, the number of data sources that feed into these estimates has also increased over time, thanks in large part to the efforts of many in this room. And in addition, the approach to extrapolation of available data has become increasingly sophisticated, also thanks to many of you. So let me show you what we think is happening with typhoid illnesses over time. Oops. Back. Yeah. So this first be reminded, of course, that the global population has grown steadily since 1980, shown in black. I won't give you all the credit for that. keeping the global population in black and now overlaying typhoid illness estimates in millions in red, and breaking all statistical rules by not showing uncertainty, a point that I'll return to soon, we see the following pattern. Please note that the 1980 estimate excluded China, which means that it's likely to be an underestimate. So if we remove China as a data point, we see this, and there's probably little point focusing on the 15-year period without estimates from 1980 to 1995. So focusing in on 1995 to 2015 brings us to this graph with global population in black, typhoid illness estimates in red. Making anything of this graph requires two major assumptions. First, that the methods are sufficiently similar to be able to compare the point estimates, and second, that the uncertainty of point estimates don't overlap. But if you do take these two large leaps with these caveats in mind, we see that typhoid illnesses appear not to be keeping pace with global population growth, but do, however, remain stubbornly present in the range of 11 to 17 million illnesses per year, depending on which estimate you look at. If we remove global population and replace global typhoid illnesses with global typhoid incidence in red and add typhoid deaths in blue, we see that typhoid death estimates are tracking closely with incidence, and this is because we've relied on a natural history approach that applies case fatality ratio to incidence to estimate deaths. This is, I think, a good point to move to extrapolation, uncertainty, and data gaps. Regarding extrapolation, I'd highlight the excellent recently published work by Marina, Antion, and colleagues who used a meta-regression approach to improve sophistication of extrapolation from areas with data to areas without data. Antion and colleagues also estimate and make transparent uncertainty, something that I've glossed over as a point in what I've shown you so far, and something that has not been described in detail in previous studies. In looking at uncertainty, they identify data gaps, including areas and age groups where more data on incidence would help to strengthen global estimates. In addition to that, we do need more and better data on disability and death associated with typhoid fever, something that we'll be hearing more about during this conference. And then finally, Antion and colleagues also identify and highlight the hidden typhoid problem in Oceania, where typhoid incidence is often very high but rarely seen on maps. As we know, Oceania has become the unsafe water capital of the world, as African and Asian regions have increased substantially their access to improved water from 1990 to 2015, shown in blue. There's been essentially no progress in Oceania. This no doubt contributes to a substantial and largely unappreciated typhoid problem in many Pacific islands like Samoa and Fiji. Okay, I'd like to finish by discussing typhoid fever in infants and children below 2 years, an age group about which we know relatively little, but we desperately need to understand more to inform vaccine policy. In this figure, I attempt to illustrate the main drivers of typhoid fever incidence and uncertainty about typhoid fever incidence in infants and young children below 2 years. First, we can be quite confident that typhoid fever incidence is not stable across this age interval. Incidence most likely describes a sigmoid curve with low incidence in the neonatal period rising with age. The main drivers of low incidence in neonates and infants include protection, disease, and lack of exposure. We understand that maternal antibody in endemic, but not necessarily epidemic settings may protect neonates. Oops. And some have suggested that disease in younger age groups may be more mild, although this is controversial. And finally, the period of exclusive breastfeeding is likely to protect infants in a number of ways, most importantly through lack of exposure to fecally contaminated water and food vehicles of typhoid transmission. Force of infection, as measured by population incidence, will have the effect of pushing the peak disease incidence towards younger age groups and the magnitude of incidence upwards. However, factors shown in orange may result in under-ascertainment of typhoid fever in younger age groups. These factors include underuse of blood cultures in neonates and infants, blood culture volume inadequacy in the young, impairing sensitivity of the test, and the higher risk of blood culture contamination in the young, also compromising sensitivity. Obviously, we'd like to understand each of these in much more detail to be sure we deliver typhoid conjugate vaccines to the correct age group to prevent illnesses and deaths. And on this note, I'd like to encourage anyone with data on the topic of incidence by month of age to please go ahead and publish them, so they're very much needed by the community. Thanks for your attention.
Thank you very much, John. So we'll keep the questions towards the end of the session.
So we'd like to welcome Cal MacLennan now to speak. Cal's from the University of Oxford. He is just started a year, also the Bill and Melinda Gates Foundation. Thanks very much, Cal. He's going to present the data from the RTSS study on invasive Salmonella disease.
Thank you, Melita, and thank you for the opportunity to present here. So, going back a few years now to 2011, the first paper with results from the RTSS AS01 malaria vaccine trials, phase 3 trials, were published in the New England Journal, and those of us, a number of us in the Salmonella community were struck by one line in one table which had Salmonella sepsis in it. And this seemed to be a strange thing to have in a paper, principally on malaria, but it just got us thinking, what's going on? So this was at an early stage of the phase 3 studies. Average follow-up amongst the two cohorts was 18 and 9 months at the time. And a back-of-the-envelope calculation in terms of what this Salmonella bacteremia might mean was approximately 500 cases per 100,000 person years of follow-up. Now, there were no details in this or the sort of 70-page supplement to this paper that would indicate anything more about the Salmonella, so this was something that we wanted to dig deeper into. So, for those of you who aren't familiar with the RTSS study, also known as the MAL-055 study, this was spread across 11 sites in sub-Saharan Africa, which was selected on the basis of having a diversity of malaria endemicity. Children were randomized into 3 groups to receive the RTSS vaccine, 3 doses, either with a booster or without a booster, or to be in a comparator vaccine group. There were 2 cohorts, which I've mentioned already. One recruited at 6 to 12 weeks of age, a second one at 5 to 17 months. And by the end of the study, which ran from 2009 to 2014, so several years beyond that first paper, median follow-up was 38 months and 48 months. Passive surveillance was conducted during the trial, and anyone who warranted a febrile admission, who was febrile, who warranted hospital admission, had a blood culture done. Whole genome sequencing of these isolates we've been performing subsequently, which is ongoing, and antimicrobial resistance profiling is ongoing as well. So, in terms of the children who were recruited, there were 2 main exclusion criteria that I want you to take note of. This is malnutrition requiring hospitalization, so severe malnutrition, and also severe anemia. And I flagged these up because these are well-recognized clinical risk factors for non-typhoidal Salmonella disease. Over 15,000 children were enrolled, and in terms of baseline observations, mean hemoglobin was 10.3, mean height for weight Z scores were 0.2, and HIV wasn't systematically tested, but any child who was unwell at the time wasn't recruited. So this is generally a healthy population. The main variable between the sites was incidence of clinical malaria, and in the analysis at the end of the study, The minimum and maximum levels were in 2 sites in Kenya, minimum in Kilifi, 0.05 cases per person year, maximum in Siaya in Western Kenya, 4.4 and 5.4 in the 2 cohorts. So our aim was to use data from this study to determine incidence of Salmonella bacteremia in children under 5 across sub-Saharan Africa. And to this end, a workshop was supported and held by the Gates Foundation and held in Nairobi back in September 2016, to which all representatives of all the sites of the RTSS trials, the Clinical Trials Partnership Committee investigators, were present. Okay, so the high-level view of this was 250 episodes of Salmonella bacteremia in all, over 50,000 person-years of observation, and the incidence following through to the end of the study was similar to that we did on the back of the envelope to start with from 2011, so 534 cases per 100,000 person-years of observation. Out of these, only around 12% was due to Salmonella typhi, so 66.5 as an incidence level. The rest was made up of non-typhoidal Salmonella, so in fact the large bulk of the incidence was due to non-typhoidal Salmonella, and this was split fairly 2-to-1 between Typhimurium and Enteritidis. In terms of all bacteremias recorded in the study, Salmonella accounted for approximately 60% of these. If we now look by site, here we have data from 8 sites. The reason why 8 instead of 11 was that 3 of the sites had no Salmonella cases at all. Out of those that did, what's clear from this is that the incidence varied considerably between the different sites in the trial, from the highest levels in the Western Kenyan sites in Siaya and Kombewa, and in Ghana, in Kintampo and Agogo, through to much lower levels at the few sites which did record levels, but at low levels, so Manisa and Lilongwe. NTS is in blue bars and typhoid in the orange bars, and again, what I hope is very clear from this was that at almost every site there was much more INTS disease than typhoid fever. The one exception is in Korogwe, Tanzania, and Dr. Lisungu will be presenting data from that site in the next session. Oops. If we split this into 3 broad age groups of 0 to 1 year, 1 to 2, and 2 to 5 years, we'll see, as expected, non-typhoidal Salmonella very much in the first 2 groups, 0 to 1 and 0 to 2, 1 to 2 years. Typhoid much more in the 1 to 2 and 2 to 5 years. And that first group for typhoid is actually only 1 case, hence the very broad confidence intervals. If we now split the data up into 6-month groups and plot that out with these large blue areas around representing 95% confidence intervals, we see the typical pattern that we'd expect for non-typhoidal Salmonella bacteremia, with the majority of cases in the first 30 months of life, with a little bit of sparing in the younger children. Bear in mind that the very youngest weren't recruited into the study. Typhoid fever, again, very little in that first year, just the 1 case I mentioned, and then the sort of rumbling level going along through to 5 years, so dispersed amongst those 1 to 5 years. And Salmonella typhimurium and Salmonella introdis, again, both of them sort of skewed to the younger age groups. So the data can be looked at on the individual level, but also on the site level. And we thought it would be useful to plot data out in relation to the sites and the level of malaria at the sites and the level of HIV at the sites, giving these spline charts. And what we've done here is we've sort of superimposed best fit lines for both invasive non-typhoidal salmonella disease and typhoid fever. So if I just sort of take you through this is slightly complicated, but what we have on both of these charts is salmonella incidence at the sites on the y-axis. On the one on the left, we have incidence of malaria from the trial at the sites plotted, and then for each site we have a blue dot for NTS and a red dot for typhoid fever. And so each site's represented by 2 dots each, and so with the blue dots for non-typhoidal salmonella disease, what we can see is sort of very strong association with levels of malaria at each site. In contrast with the red dots and the red line, very little association, no association really at all with typhoid fever, and this is in keeping with data that John Crump's published from sites in Tanzania. In contrast, it's a little bit more complicated with HIV on the chart on the right. As I mentioned already, HIV wasn't systematically tested for in the study, so we've had to use population levels for adult HIV prevalence at the different sites. On the right side, the Western Kenya sites, high levels of non-typhoid, of Salmonella bacteremia, high levels of HIV. But on the left-hand side of that chart, we've got the West African sites, so Ghana, Burkina, again, quite high levels of Salmonella bacteremia, but low levels of HIV. This is a very uncooperative clicker. Okay. In terms of outcome, what was actually quite striking from the study was the low level of mortality. There may be several reasons for this. On the figure on the left, we've got the mortality following Salmonella bacteremia. Only 8% of the cases of non-typhoidal Salmonella bacteremia resulted in fatality. So few cases of typhoid fever make this somewhat difficult to interpret, but there's only 2 fatalities in typhoid fever. What we have in the green line below that was actually death from other bacteremias, which was rather greater. But if we look at the chart on the right, the green line and the green bar is from all deaths throughout the study, from all study participants. It just indicates that these were generally children who did rather well. You've got the typhoid and NTS lines underneath that in red for typhoid, blue for non-typhoidal Salmonella bacteremia, and certainly for NTS bacteremia, there's a significant difference with the general population in the study. So that, I guess, brings us to the question of why was the case fatality rate somewhat lower than what we've expected from previous studies. Well, children had access to healthcare facilities, access to blood culturing and good treatment, so that might be part of the reason. But again, the fact that we've recruited rather a healthy population of children might have a bearing on this as well. So how about the association with malaria? Given the effort that was put into this study was largely on malaria, it allows us to interrogate the findings in relation to malaria quite nicely, particularly as this was a longitudinal cohort study. So in fact, over 80% of the cases were aparasitemic at presentation. However, looking at those that had malaria in the 2 weeks prior to their onset of Salmonella bacteremia, a lot more of the children with NTS bacteremia than those with other bacteremias had a positive malaria parasite slide, again in keeping with findings for others. What the study did allow us to do was to look at children over the whole time course of the study and then see how many episodes of Salmonella bacteremia how many episodes of malaria they had, and how much Salmonella bacteremia. If a child had no cases of malaria, their incidence of Salmonella bacteremia was relatively low, 136 per 100,000 person-years of follow-up. As you ramp up with an extra malaria episode per year, that incidence level almost seems to double with each step down that line. And these differences in terms of incidence ratio compared to the reference level of naught infections is highly significant. In terms of the RTSS vaccine itself, there was a non-significant reduction in incidence in the group that received RTSS, so 0.83, but with the confidence intervals this was non-significant. And this might be in keeping with what we expect of the burden of NTS, the contribution of malaria to NTS burden. So in conclusion, Salmonella is, from this study, still a major and persistent cause of bacteremia in sub-Saharan Africa. The three commonest serovars are Typhimurium, Enteritis, and Typhi. But I've highlighted already, INTS disease was much more common than typhoid fever. A monovalent vaccine, we've got typhoid vaccines obviously coming through, could have presented 12.5% of bacteremias from Salmonella. But if we had a trivalent vaccine that covers Typhimurium and Enteritis as well, we'd be able to cover over 90% of cases. So such a vaccine would be able to have a major public health impact. There are a huge number of people to thank. I'd particularly like to highlight Simon Kariuki on the top left here, who was the lead investigator from the CTPCI who contributed to this, and to Ryan Wygand and Nellie Westerkamp from CDCs who've helped with the analysis. And thanks to the Gates Foundation for funding. Thank you.
Thank you very much, Cal. It was wonderful.
Our next speaker is Sam Kariuki from the Kenya Medical Research Institute, and he will talk to us about enteric fever outbreaks in Africa. Sam.
Thank you very much to the organizers for giving us this opportunity to share this data. I'm going to talk about enteric fever in Africa, and we've just got a very excellent exposure there by John on the epidemiology of typhoid globally. As an introduction, we note that there are very few population-based data, especially on incidence and mortality in Africa. And we're really grateful to the Gates Foundation for initiating the SETA incidence study on typhoid in Africa. And this is going to give us a lot of data on some of the missing gaps, as we've heard from John's presentation. We do get data from hospital-based studies, but these, again, are far and in between. Most of them are really sentinel sites surveillance data, which does not give a whole picture for estimates at the global level in Africa. We find a lot of NTS in West Africa, in Central Africa, and East Africa, but we're now increasingly finding more cases of typhoid, especially from informal settlements, and we'll see this much more in my report as I go on. And we do not understand particularly the regional differences, especially from country to country, from region to region, in the African setup. In Africa, especially in sub-Saharan Africa, we have huge problems, especially with overcrowding and sanitation, especially with populations moving from rural areas, coming to the cities, looking for jobs, and they end up in informal settlements where you find affordable accommodation for these workers. And this is a picture of one of the sites east of Nairobi, about 20 kilometers east of Nairobi, where we're trying to map NTS and also typhoid hotspots with a view to finding out if we can be able to intervene with vaccines at the hotspots. And you find population density extremely high, and this kind of environment, of course, is very, very ripe for incidence of typhoid being extremely high, and with very frequent outbreaks. This is a map that many of us have seen with areas highlighted in deep red that are endemic and hyper-endemic regions in the world. And you can see that Africa is disproportionately presented here, and lots of issues, especially with growing population densities in cities. And I'll quote extensively from this work that was done by a colleague from University of Cambridge, Vanessa Wong, who did her PhD under Gordon Dugan's laboratory. And she did a fantastic job, a global collection of the genomics of Salmonella typhi. And again, we'll be looking at aspects particularly that touch on Africa. And in Africa, especially sub-Saharan Africa, we find a lot of typhoid clusters in many, many regions now, reporting from, you can see West Africa, Central Africa, East Africa, South Africa. Virtually all these regions are reporting both the epidemic haplotype 58 clade and also the non-H58 clade. But increasingly, you find that most regions now have the H58 clade that is multi-drug resistant and rapidly, very rapidly spreading to many, many regions in Africa. The reason why I've searched the publications or MacLennan and brought up this list of publications is to let you know that in the last 10 years or so, we're getting more and more data out of Africa showing high incidence of Salmonella typhi, and much more so, especially for multi-drug-resistant typhoid, which poses a major challenge in treatment of this disease in Africa. West Africa, South Africa, a number of publications, very, very recent ones, and even in Central Africa, and all of them having the same, trying to go back, but yeah. All of them having the same issue, especially of multi-drug resistant clades rapidly spreading, especially in informal settlement areas that this research has gone on. Here in Uganda, recently, as you may remember, we had a large outbreak of typhoid fever, and we had reports of acute typhoid infections causing perforations as well. So we're getting more data out of Africa than before, which is very, very important, and this will drive policy, especially towards implementation of typhoid vaccine for prevention. As I said, we had recently this very fantastic work that was done by Vanessa Wong during her PhD, and the genomics of Salmonella typhi was very well elaborated, especially for the African continent. And you find, of course, that the H58 epidemic clade that originated from Southeast Asia rapidly spread to the rest of the regions, especially in Africa, through the intercontinental trade and movement of people. And within a very short time, we have this clade now well spread out in many, many areas, especially in Africa. And you see, in Kenya, where it first landed, and then we have the same clade now spread to Tanzania, Malawi, and even South Africa. And when you look at the African continent, we first reported about this clade way back from 1994, when suddenly we realized that our strains, previously between 1988 and 1994, had been most often susceptible to most commonly used antibiotics. But from the year 1994, we started realizing that about 60% of isolates we were receiving in the laboratory were multi-drug resistant. And in fact, they reduced susceptibility also to the quinolones, especially to ciprofloxacin. And we're able to describe, again, with collaboration from Gordon Dugan's lab, that indeed the local strains had been replaced by this epidemic-clade haplotype 58 that was also highly endemic in Southeast Asia. And then this then was the beginning of the hub of H58 spreading to other African countries, starting from the East African region, going to West Africa, and then to South Africa. So what are the major challenges in tackling typhoid in sub-Saharan Africa? And these are issues that have persisted over time and which hopefully we'll be able to together manage. If you look at diagnosis, typhoid is a very difficult disease to diagnose clinically because really we do not have any clinical symptoms that per se spell out that this is typhoid that you're looking at. But then clinicians will depend on high index of suspicion and the knowledge about the local epidemiology of the disease, and of course, looking at the clinical history and presentation as well. Looking at the laboratory diagnosis, again, huge challenges, especially in areas where we have challenges of infrastructure, especially in Africa. Blood culture or bone marrow culture, which is really the gold standard, requires a lot of infrastructure investment. And very few laboratories will be able to have this in place. And much more so, especially because it takes a couple of days before you realize that you're actually being able to diagnose typhoid fever. If you look at serology, it's only useful from week 2. And the window test that has been used over the last 100 years or so has huge problems of implementation, especially with false positive test outcomes, especially in this region where the disease is endemic. The stool antigen test, only useful after week 2, again, huge problems, especially with huge background of antigens in endemic areas. Unit culture, nearly a month. But increasingly, of course, there's a lot of research that has gone into utilization of PCR as a rapid test, especially that will mirror clinical diagnosis. Whole genome sequencing and metabolomics, these are new technologies that we hope will be able to come into play and be used as bedside diagnosis, especially now that we know that we can be able now to match whole genome sequence outcomes with some of the phenotypic characteristics of the disease outbreaks. And recently, 2015, this work that was done in Zimbabwe, trying to look at 2 tests, the accuracy of 2 tests, Tubex typhoid test and on-site typhoid IgM combination tests, and comparing that to the gold standard, you can see that the sensitivity and specificity of both are fairly acceptable. And obviously also, these are tests that are easy to adopt in many of the rural areas in Africa. And so long as we are able to adopt this and be able to bring the prices down for this kind of kits, then we can adopt them as rapid diagnostic kits that could easily really revolutionize the way we diagnose typhoid in the region. We need to look at issues of treatment. We have huge problems in Africa. We're coming from a region where antibiotics are easily available over the counter, hence contributing a lot to multi-drug resistance, not of typhoid only, but many other infections. And for multi-drug resistant infections, we use fluoroquinolones quite often in the region to try and treat typhoid cases. And ciprofloxacin especially, which is commonly available, is very, very effective, especially for fully susceptible strains. But when you find multi-drug resistant strains, which is sometimes in some regions over 65% of the strains we receive in the laboratory, then we can only apply the later generation of fluoroquinolones, gatifloxacin as an example. Azithromycin has been used quite widely and is very, very effective. But again, we are seeing signs that resistance is cropping up. The third generation cephalosporins and the fourth generation ones as well, they are reliable, but these mainly being used for hospital admitted cases, not in outpatients. As far as vaccines are concerned, this is really an area that John has touched very well, and there are several WHO recommended vaccines. I think the major problem with the implementation of vaccine programs in Africa is that policymakers rarely look at typhoid disease as a major threat to population public health. And the data that's coming out now will emphasize the need for policymakers to look seriously at how we tackle typhoid outbreaks, especially now with increasing population densities in huge cities as people come to seek jobs in these areas and end up especially in informal settlements. Prices are still high because these vaccines are not widespread in use. Most often we use them for travelers and also those working in the hospitality industry. So it's really an issue that we need to emphasize to governments in Africa to be able to prioritize use of vaccines for prevention. And some of the work we are doing in Nairobi, especially mapping hotspots for typhoid fever, again emphasizes this issue for policy. So in conclusion, there will always be increasing population densities, especially in informal settlements in Africa, as people come to seek jobs in big cities. So it's very, very likely that this problem will be with us for some time to come, and we have to prepare how to tackle it. We've seen that accurate diagnosis is a major problem because this is not a disease that has symptoms that per se spell out typhoid. We need to adopt simple, affordable, rapid kits that can be deployed under field conditions and also as bedside diagnostic kits. Burden of disease data and economic implications are important to document, and this is what moves governments to implement policy, especially on vaccination. We've seen that resistance is increasing, especially to most of the commonly available antibiotics, including fluoroquinolones, which are the drugs of choice in Africa. Again, this is a huge problem. But of course, in the long term, improving hygiene, clean water supply, and dealing with overcrowding and sanitation will be the long-term goals to be able to control typhoid. So I'll acknowledge many groups that have been involved in this work, from Kenya Medical Research Institute, the Aga Khan Hospital, the Sanga Institute, especially Gordon Dugan's group, and also all those that have been involved both as clinicians and also in field work, getting this work together. And together in Africa, we'll make this disease controllable. Thank you very much.
Thank you very much, sir.
Okay, to close the session, I'd like to invite Nick Feasey to come up and speak, please. Nick is at the Liverpool School of Tropical Medicine and has also worked for 6 or 7 years, I think, in Malawi. Welcome, Nick. Thanks.
Hello, thanks to Melita for the introduction, and thanks to the Coalition for the invitation to speak and to give an overview of strains of Salmonella associated with invasive disease. So the first slide, I'm going to talk about the different bugs. That Salmonella typhi is associated with invasive disease is hardly news, but the picture with non-typhoidal serovars is rather more complex. This cartoon illustrates the conventional wisdom that Salmonella typhimurium and Salmonella enteritidis are conventionally thought of as generalists. They have a diverse genomic repertoire supporting a broad host range, and they're typically associated with enteric or diarrheal disease with a low mortality. There's a number of serovars, such as Dublin or Cholera suis, which are more host-adapted, but when they get into humans, they are more invasive. And then on the far side, you can see the host-restricted bugs, that's Salmonella typhi in humans and Gallinarum in chickens. These have genomic degradation of specialist genes with specific additional virulence genes. And they're associated with invasive disease and high mortality. My turn to run the gauntlet of the clicker. There we go. The picture in sub-Saharan Africa has always been more complex, and there are numerous case series of pediatric invasive non-typhoidal Salmonella disease in the pre or early in the ART era. So from Nigeria in the 1970s, and Kenya, Gambia, DRC, and Rwanda in the 1980s. And of course, in the early 1980s, as HIV started to emerge, everything changed. Come on. Is the battery going, perhaps? Can you click the next slide, please?
I think it's the lower button.
I was pressing the lower button, it just wasn't going. So, yep, that was the right slide. Thank you. So, in the early 80s, everything changed with the emergence of HIV, and INTS disease was one of the first AIDS-defining illnesses. And in John Crump's systematic review of bloodstream infection in Africa, it was no surprise that INTS disease was one of the most common causes of bloodstream infection. So the rest of my talk will focus on the biology of the bugs, and this slide is just in here to remind us, or to remind me, that Gordon Dugan and I came under criticism from Mark Ackman 2 years ago in Bali for not explaining our phylogenetic trees well enough. So what follows is a series of models comparing specific serotypes of Salmonella predominantly using maximum likelihood methodology, using whole genome sequence data and comparing single nucleotide polymorphisms. This is a baobab tree. It gets harder. So next slide, please. So back in 2007, 2008, Rob Kingsley and Chisomo Msefala sequenced 30 Salmonella typhimurium genomes from Malawi and from Kenya. And they identified a novel multi-locus sequence type. So this is just an MLST tree based only on 7 housekeeping genes, but that small snapshot of the genome identified something fundamentally different about the African Salmonella typhimuriums, that they belong to multi-locus sequence type ST313, which is different from the ST19s, which tend to cause diarrheal illness in resource-rich setting. They also noted genomic degradation in the genes responsible for an enteric lifestyle, and genomic degradation in genes or in pathways which are degraded in Salmonella typhi. And this is a pattern seen in a number of other pathogens becoming host-adapted as well. They detected a novel prophage repertoire and a multi-drug resistant cassette in the virulence plasmid. As sequencing became cheaper and more feasible to sequence lots of bugs, Chineri Okoro looked at 179 Salmonella typhimurium isolates, and she identified that there are 2 lineages of highly related Salmonella typhimurium isolates which appear to be relatively restricted to sub-Saharan Africa. These bugs were all ST313s. Again, they have the novel prophage repertoire, and again, they're multi-drug resistant. Although critically, in this study, the lineage 1 isolates did not contain chloramphenicol resistance, whereas the lineage 2 did have chloramphenicol resistance. And numerous groups have taken on this story to investigate whether there is a fundamental difference in phenotype between these groups of isolates, between the ST313s and the ST19s. This slide is from a recently published paper by Sean Owen from Jay Hinton's group, looking at the novel prophage repertoire. The first thing to say is that 3 of the prophage regions in ST313 are the same as those present in ST19, but all 3 are defective so that they're not expressed. But there are 2 completely novel prophage regions, the so-called Blantyre type prophage 1 and BTP5. BTP1 is fascinating in that it's got the highest spontaneous induction rate ever seen in prophage biology, which is interesting, and it also contains the putative virulence gene BTP1. So again, there is the signature of something different going on in the African ST313s. Next slide, please. What follows is a whirlwind summary of a number of papers which have come out exploring these differences. So there's a paper on this ST313-DTD gene. The ST313 stimulate less inflammasome activation than ST19. This is an interesting paper which suggests that at a cellular level, the ST313s are less invasive than the ST19s, but more so than Salmonella typhi. And one of the things this illustrates is that people doing fundamental science use the term invasive differently to clinicians and epidemiologists thinking about bloodstream invasion. There was the Ramachandran paper saying that ST313 with naturally attenuated flagellin elicits reduced inflammation and replicates in macrophages. And more recently, that the ST313s display loss of multicellular behavior. This paper speaks to the possibility that ST313s and ST19s are behaving fundamentally differently in the environment. And this is very important because it raises the possibility that there is an environmental niche that these bugs are living in, which can be targeted through suitable water sanitation and hygiene interventions. And most recently, there's a paper on pseudogenization of the secreted effector gene SSEI, which confers rapid dissemination. At the time of the original paper by Chisomo and Robb, it was speculated that perhaps African ST313s are in fact host-restricted like Salmonella typhi. So some of the ST313s were put through chicken models and in fact found to be more invasive in chickens than ST19s. So that hypothesis was rejected. But again, this highlights the different behavior of ST313s when compared to ST19s. More recently, the plot has thickened somewhat. So Public Health England are now sequencing all of their Salmonella genomes, and they have sequenced 3,000 Salmonella typhimuriums over the last few years, and they've identified that 79 of 2,888 were in fact ST313, and this was a surprise. You can see the two red lines, back a slide please, if you can. Oops, sorry, that's me now. That seems to be working better. It's on a hair trigger. There we are, stay. Right, so you can see the red lines, lineage 1 and lineage 2 are those isolates which those lineages which Cherian previously identified. And around them, however, there is a cloud of diversity. So there is a huge diversity of ST313 represented by the PHE collection. And when you drill into those bugs, they are associated with travel to Africa, multi-drug resistance, and conservation of the prophage repertoire, novel prophage repertoire, whereas the remainder are isolated from stool, they have prophage diversity, and they're drug susceptible. So that again illustrates the limitations of MLST typing, just looking at 7 housekeeping genes rather than looking at the entirety of the genome. What's going on with Salmonella enteritidis, which is the second most common cause of INTS disease in sub-Saharan Africa? So in this study, I looked at 680 Salmonella enteritidis genomes, and there are some parallels with the typhimurium story, but it's not identical. Once again, we can see some novel clades emerging, which appear to be restricted to sub-Saharan Africa. So the orange bar at the bottom, you can see a West African clade, and here a Central and East African clade, and here is a global epidemic clade which is associated, this is the Edwina curry, associated with eggs and poultry. But the way in which enteritidis differs from Salmonella typhimurium is that there are lots of African isolates in the global epidemic clade as well. So 2 types of enteritidis, invasive non-typhoidal Salmonella disease, are occurring side by side. The African clades again have the novel prophage repertoire and again associated with multi-drug resistance. Again, we put these bugs into context of the Public Health England collection. Here, 3,000 Salmonella enteritidis genomes, and we didn't find the African enteritidises in the Public Health England collection particularly, just 7 isolates this time, 6 of the West African and 1 of the East African. So again, there seems to be something about a specific niche in sub-Saharan Africa which facilitates the emergence of these bugs as major pathogens. Moving on to Salmonella typhi, Sam has already touched on this story, but there is a novel clade of Salmonella typhi as well. And as Sam mentioned, just about the time of the last meeting in Bali, Vanessa Wong's excellent paper was just being released. And she described the emergence of the H58 lineage, or as she has more recently called it, the subclade 4.3.1. I will leave it to you to decide which is the more catchier of those, but I encourage you to read Vanessa's excellent work. She looked at 2,000 Salmonella typhi genomes, identified the emergence of this clade, which is associated with multidrug resistant and diminished fluoroquinolone susceptibility. In this last map, this model uses Bayesian mathematics rather than maximum likelihood, and it incorporates whole genome sequence data and date of isolation and place of isolation as well. And in this model, it's estimated that the H58 clade emerged about 30 years ago in South Asia and has disseminated around the world, arrived in East Africa some time ago. And that fits with what we've seen and what has been reported in terms of multiple outbreaks of typhi in East and Southern Africa over the last 10 years, from Uganda, Zimbabwe, Zambia, and more recently, Malawi. So one thing that all of these strains associated with invasive disease appear to have in common is a propensity to acquire antimicrobial resistance. And this has always been a problem, but more recently what has been emerging is a story of extended spectrum beta-lactamase producing variants. And this is, in many contexts, in Africa in particular, the doomsday scenario. So BLAST-CTXM15 is the gene globally which is most strongly associated with cephalosporin resistance in E. coli and Klebsiella. This was first reported in Salmonella typhimurium in Malawi in 2009. Shortly afterward, it was reported in the same plasmid in one of Sam's reports in Kenya. And as Cal has highlighted, we've got lots of bugs emerging from West Kenya, and recently up to 57% of Salmonella typhimuriums from West Kenya were found to be ESBL producers. It's also an emerging problem in Salmonella typhi, and I know that people are going to reflect on this. But this has the potential to make Salmonella untreatable in many contexts, the emergence of cephalosporin resistance. So in summary, there are novel clades of multidrug-resistant S. typhimurium and S. enteritidis exploiting the high prevalence of immunosuppressive conditions to cause epidemics of invasive disease. Whether it's more invasive or less invasive is not something that we can easily modify, but whether the genomic signatures reflect a niche adaptation to a particular environmental reservoir is something that we might be able to target and exploit. MDR H58 S. typhi has established itself globally, and drug resistance across all of these pathogens is a major and emerging problem, and the ESBL is particularly frightening. Oops. I'm going to finish by advertising a study that my colleague at the University of Liverpool, Jay Hinton, is conducting. Jay has the funding to sequence 10,000 Salmonella genomes. He is interested in understanding the epidemiology, transmission, and virulence of invasive clades. And if anyone would like any of your INTS isolates sequenced for free, he would be delighted to do it. Blanca, can you stand up, please? So if anyone would like to get your isolate sequenced, please speak to Blanca, who has a poster later on. I'm just going to conclude by thanking the Coalition again for the invitation, and also to thank my funders, the Gates Foundation and the Wellcome Trust. Thank you very much.
Thanks very much, Nick. Wonderful whistle-stop tour there.
Thanks to all the speakers for being very good about sticking to time, so we do have some time for questions and comments. So I'd ask each of those who are asking questions to please state their name, the institution and country they come from, and if possible, indicate who their question is directed to.
It's just the bit where you come to the microphones, people.
Yeah.
Thank you very much for an interesting series of presentations. Eric Mintz, CDC. I was wondering if you could give us a little more hints about that mysterious environmental reservoir where these untreatable INTS clades are living. Have we ruled anything out? The usual suspects, chickens, cows, and do we have any sense of what sort of conditions they survive well in that might inform, where we should look for them or how we could intervene? That's for you, Nick.
Thanks, Eric. The short answer is no. I don't have any answers for you today. This is the subject of some research which is just starting that is funded by the Gates Foundation, work that I'm doing in Malawi and Christine Moe is doing as well. There is a pilot study which is being conducted in Malawi, and Melita will be giving a talk on that in the late breaker session, so I won't steal her thunder. But nothing revolutionary as yet.
Thank you very much. I'm Narendra Arora in Clint, New Delhi. My question is also directed to Nick. It's my ignorance, but I would like to know whether any of the Salmonella species undergo mutation in sewage and the genetic structure changes over time while it is in this environment.
So these isolates are highly conserved, so they are undergoing evolution. Precisely, I don't know as yet, because that's just that work is just beginning now. I think what's more interesting is what behaviour they have in sewage. So there is the concept of a viable but non-cultivable state. One of the things we're interested in is exactly what the nature of their metabolism is when they're in a viable but non-cultivable or environmental state. So I can't answer that question at the moment, but the work is ongoing.
Sophie.
So, thank you. That was a very informative session. Zulfi Butta from the Arcon University and in Toronto. So I have two questions. One pertains to the last comment, which is, what is the viability of using sewage surveillance, just like we do for polio surveillance in many endemic areas, as a means of looking at trends or, the proportion of drug-resistant isolates for both Salmonella, human Salmonella, as well as Salmonella enteritidis. And the second question that is related is, this doom and gloom scenario of these clades kind of spreading all over the world and making a disease completely unmanageable. How does one balance that out with the observation over time that some of these multi-drug resistant isolates also have limited survival in the environment? I mean, they may be associated with the greater virulence, but they do tend to go up and down, and certainly with the multi-drug resistant isolates that we saw in the 80s and 90s, they went up to about 70–80% prevalence, and then they came down to a lower level of stability. So how much do we know from history and ecology that our predictions in terms of these clades becoming universal are true?
So I think your suggestion of more environmental surveillance and particularly sewage surveillance is an excellent one, and it's one of the things that we propose to do in a number of sites. I think there's a huge amount that we don't know about the environmental persistence of both typhoidal and non-typhoidal strains, and partly that's because it's been very difficult to culture them. There have been very few outbreaks where an environmental source has been identified in recent years. So I think there is some very basic work to be done optimizing techniques to isolate the bugs from the environment. In terms of how you marry up environmental persistence to pathogenesis and drug resistance, I think it's difficult to draw conclusions from that until we've actually successfully isolated it from the environment. But your point is well made that drug resistance does appear to wax and wane. There does seem to be this trend, particularly in the NTS, towards ever greater drug resistance, and we haven't seen regression of the MDR state, and in fact we're seeing moves towards cephalosporin resistance in the typhimuriums and enteritidis. With typhi, as you say, the picture is much more dynamic and different in different pictures, in different settings.
Question.
Thank you for a nice session. Iruka Okeke, University of Ibadan. Question is for Cal MacLennan. You talked about lowered mortality in the study that you described, and I just wondered whether there's any information on the nutritional status of the population in the study, as well as the few that did die.
Thank you very much. Is that working? So, nutritional status in the study. These children were essentially, there was, as I mentioned, severe malnutrition was an exclusion criterion. So these were generally more healthy children than those that we might typically find winding up presenting with particularly INTS disease. In terms of the nutritional status of the actual cases who did die, what we're now doing with the analysis phase is taking advantage of the longitudinal data that we have available from this study to analyze the individual cases in more depth, and that's something that we will do as part of the ongoing analysis. But I think, if anything, the fact that children with severe malnutrition, children with severe anemia, children with underlying illness were excluded from the initial recruitment into those cohorts would suggest that the incidence levels that we have here are a minimum one. So you're naturally excluding sicker children with clinical associations that are known to co-associate with particularly INTS disease. Thank you.
Middle microphone, please.
Thank you. This is Rick Malley from Boston. Thanks to the panelists for great talks. Cal, this is a question for you. There's an interesting hypothesis in the RTSS trial that the reported increased mortality in the malaria group in the vaccine group may in fact be a misinterpretation of a lower incidence of disease in the rabies control group. This is a hypothesis that Brad Gessner and Adam Finn have brought about. I wonder if that hypothesis were in fact supported, that could in fact mean that your reported rates of non-typhoidal salmonella may in fact be a slight underestimate, because you would expect some protection in the RTSS group, and if rabies vaccine provides non-specific protection against a host of diseases, it may also provide protection against non-typhoid salmonella.
Thank you very much for bringing up that interesting question. So Brad has been in touch about this and asking the question of whether this might be borne out in terms of the bacteremias. So there was a decreased incidence of, I think, meningitis as well in the rabies group. So just to explain to the general audience who might not be as familiar with the RTSS study, the two control groups had a different control vaccine. The older cohort had the rabies vaccine, the younger age group had a meningococcal C conjugate vaccine, and there was what appears to be less meningitis and cerebral malaria in the older control group. So the question was, might this be borne out with the bacteremia as well? I haven't presented that, but in terms of the Salmonella bacteremias, there wasn't a difference. So we have looked at that at the request of Brad. In terms of all of the bacteremias, that's a little bit more difficult to look at, because what's a pathogen, what isn't a pathogen, hasn't been sort of carefully gone through at the moment with that. But certainly in terms of the Salmonellas, it doesn't look like that effect has been translated through, but thank you for the question.
Okay, thank you very much. We've been told to be very fierce with time, so we're going to wrap the session up. Who are your questions for? Nick. Could I encourage you to grab the speakers right now and chat with them about their talks? Thank you very, very much. But we are going to run to time, otherwise I'm going to get in trouble. Few business things. First is to encourage you to please go to the posters. All the posters are up all the time. Poster presenters will definitely be by their posters this afternoon, but I'd encourage them to get to their posters now, so please do that. And the second thing is just to repeat the invitation to the TIVAC reception, which is open to everybody at 5:30 tomorrow, straight after the human-controlled infection session. Thanks very much, everybody.